Yes.
All right, we are being recorded. I will launch right into it if we are ready. It's so nice to meet you all, and thank you so much for the opportunity to speak with you tonight. We're very excited to give you a talk about our work. I am, as our speaker introduced, Ingrid Rochon, and this is Faridah Dahlan. And I'm going to start us out tonight talking about when birds collide. So who are Faridah and I? We are members of the Smithsonian's Feather Identification Lab. We are a lab of 5 people. That's Dr. Carla Dove in red at the front, Faridah and myself. We also have Jim Watton and Sarah Luttrell. We are a working group that is embedded within the National Museum of Natural History's Division of Birds. We work on the National Mall, up on the very top floor of the Natural History Museum building within the collection, which looks exactly like this. These hundreds of old wooden cases filled with about 500,000 preserved bird skins and skeletons. It is the very best place in the world to be if you want to understand avian biodiversity, and the uses of the museum's collections are innumerable. They are visited every day, every working day of the year by scientists who are studying comparative anatomy, ecology, disease, bird conservation. We have the largest collection of bird skeletons in the world, which is a great resource for zooarchaeologists. Artists, especially field guide artists, find our collection an invaluable resource. All of those paintings in your Sibley guide are based off of museum specimens. We have an alcohol collection that can preserve DNA that can be used to study zoonoses, past and present. We have extinct species. There are people even dissolving feathers to find forever chemicals, the things like PFAS and plastics that are in the environment and find their way into birds. Every one of our specimens is a source of genetic material, even if they weren't collected with genomics in mind. And then there's what we use the collections for, forensic identifications. And what in the world could possibly be going on that you need a full-time laboratory of 5 people doing bird identification alone 24/7, 365. Bird strikes. Each year, bird aircraft collisions, which we call bird strikes, cause an average of $44 million in damage to US Air Force planes and about $600 million in damage to US civil aircraft. And that's damage that can look like this. You're busted in radomes or nose cones, shattered windshields, damaged sensors, even whole aircraft loss. Many of you may have seen this on television when it happened. US Airways Flight 1549, the emergency landing, better known as the Miracle on the Hudson. This was obviously a full aircraft loss and a very near miss of many people could have been seriously injured or hurt in this case. This was caused by a collision with a flock of geese. Incidents like this are not only costly, but potentially deadly, and they are not a new thing either. The first bird strike was reported by Orville Wright in 1905. He struck a small bird while he was making passes over a cornfield in one of the Wright Flyers. And at this point in 1905, they had only just figured out how to steer the airplane. They had it in the air for 5 minutes at a stretch. 5 minutes was about the record, and they are already hitting birds. We do not know what kind of bird they hit, but it is not too far into the future when people begin to realize that knowing that might matter. In 1912, the first bird strike fatality occurred when aero pioneer Cal Rogers collided with a gull, which then became jammed in the control cables of his aircraft. He crashed at Long Beach, California, was pinned under the wreckage, and drowned. And this is maybe one of the first incidents in which it becomes a little clearer that it does matter what type of bird you are hitting. This early aircraft was not designed to withstand a hit with a gull, a large, relatively large-bodied bird, and come out unscathed. But it was not until 1960, with the rise of mass commercial air transport, that people really started paying attention, and people started paying attention because of this incident. On October 4th, 1960, 62 people were killed when Eastern Airlines Flight 375 crashed out of Logan Airport into Boston Harbor. Within a few minutes of takeoff, eyewitnesses reported seeing smoke and flame shoot from the engines shortly before the plane nosedived into the water. Later, at the end of the runway, investigators found hundreds of bird carcasses, and when they pulled the airplane up from the bottom of the harbor, they found that the engines were completely clogged with feathers. The remains were sent to a woman named Roxy Laybourne, who was working as a bird preparator and a technician, just like I started out at the Smithsonian, and she was able to match up those feathers to the specimens in the collection. She determined that they belonged to European starlings. A flock of them had been sucked into the engine, and so the mass of that entire flock going through the engines took out 3 out of 4 of the plane's engines, and they were determined to be responsible for that crash. And Roxy Laybourne had been so instrumental in figuring out what happened that the Federal Aviation Administration, the FAA, kept sending her bird remains and drawing on her expertise whenever it was needed, which we continue to do today. Roxy Laybourne's legacy is our lab. She mentored Dr. Carla Dove, who is now my mentor. And our lab, which originated in Roxy Laybourne's work, continues to investigate over 12,000 bird strike cases per year. So we're very busy. Before you get worried, most of those 12,000 cases are harmless, they are not fatal, they are not even damaging. But we're going to talk to you a little bit today about how we investigate those cases and why we investigate every one of them. We're going to start out with our methods, because I think that's what's going to interest you most as birders. Just being fascinated with birds is what we do. And we'll end with the fruits of our labor, how this information is used to keep you safe. We use 4 different methods of identification to identify bird remains. First, we can identify whole birds or recognizable parts of birds that are found dead on runways using photographs. That's easy enough. But we don't always get the whole bird. We are the feather identification lab. We use whole feather matching to match up feathers to museum specimens, just like Roxy Laybourne did way back in 1960. But a lot of the time, we're not even getting feathers, and our technology has advanced. As Faridah is going to talk to you about today, we have a third method of molecular identification. We can take samples of bird DNA, we can barcode that DNA and identify it to species, and then finally, we can look at the microscopic feather structure. For even stuff that's been blasted into smithereens, we can take the dust and look at it and be able to tell you what bird that is. We rely a lot on our molecular and our microscopic identifications, because most of what we receive in the aftermath of a collision is what we call snarge. And Faridah, since you're the queen of snarge, could you tell us a little bit about it, please?
Certainly. Bring on the slide, Ingrid. What is a snarge? The word snarge has been credited to our lab, even though Carla said we were not the first one to have coined the word. It was just a word to describe that bird preparers call things that come out of birds when they prepare the birds for the museum. But somehow we got we got the credit for it, and we may not have coined the word, but we use it every day in our work. We use it when we give presentation like today. We get when we give tours and all those other things. And we have certainly popularized the word, and we even adopted the word snarge for a lab sticker that we will show you. There we go. It says God snarge and please report, because that's what we want. Anything that resemble remains. And we recently found a poem called Snarge, and it was quite lovely. They mention the museum, they mention the Smithsonian, the collection, and they call us wreck site investigators. Even though we never go to the to where the accidents happen, it's just for us, we just get everything through the mail. But the poem is quite lovely. It's a recent poem, and the star of the poem is Stunus vulgaris, starlings. And so if you get a hold of that poem, it's a silly poem, but not too silly, but it's quite quite lovely. Next slide, Ingrid. Thank you. So we received different kinds of snarge. I'm not sure whether we can, if snarge can be pluralized. Is there snarges? We've never used the word in a plural form. We always say snarge. So I shall not say snarges then. We received different snarge. I can't quite see the slides, but we get feathers, we get tissue, and unfortunately, sometimes we get irradiated stuff, especially after September 11. If you send it to the wrong address, we will get really crispy samples that you can't extract DNA successfully from it. And then that's when we give it all to Ingrid to try and ID it. And if it's moldy, like recently we have Ingrid had a bag, a Ziploc bag with 16 birds in a soupy bag, but she managed to take every one of them out. We managed to get DNA from all 16 birds, and we managed to get ID to species for all 16 birds. So we tried to work really, really hard to get IDs for whatever people sent us. And it doesn't matter how the snarge look. We will process everything that we get. We encourage folks to send whatever samples they have when there is a bird strike event. And of that 12,000 samples that we process, there is a lot more that is not reported. So 12,000 is keeping us busy quite a bit. And that's it. Back to Ingrid. And there you go. Please report your snarge.
This is snarge. We're going to go into each of our 4 methods to show you how we turn these smears and globs into bird identifications. But since I don't know, maybe some of you are molecular specialists, but I was led to believe you were birders. So I'm going to start you off easy. You have all identified birds from photographs. That is just birding without relying on your field marks, without being able to hear the bird call, which I really wish I could. When I get an Impidonex flycatcher, and I really wish it could still call, but we work with what we've got. But our first 2 methods, photographs and whole feather matching, are very intuitive. Can you identify the bird pictured here from just one of its feathers? I bet you can. Take a minute. What North American bird does this feather belong to? You can type your answers in chat if you wish. I can't find the chat. Or you can unmute yourself and holler out the answer. I'm going to try and find the chat. There we go. Yes, I see the correct answer from Barry. That's a red-tailed hawk feather. Easy. Cat. My cat is being a nuisance. Get out of here. There you go.
No need for DNF that time, Barry. We got it. You got it.
All right, what about this one?
Morning, Doug.
Hey, you've done it. If anybody had a moment of pause, this matches up to the museum specimen very nicely. This is exactly how we do it. All right. A little bit harder. What's this one?
Cedar waxwing.
No. It looks a little like the red-tailed hawk, but it is not a baby red-tailed hawk. I'll give you another feather. Same bird. Anybody?
I may still have a job.
I
Maybe a cast.
think I heard it. American kestrel. And as you'll notice, the male has a different tail than the female. So these are the sorts of things that we need to take into account when we're doing this whole feather matching. We're going to need to use the collection to look at birds of different sexes, different ages, different molts, and we need to develop an awareness of the range of individual variation. Kestrel tails have lots of individual variation, but they all follow the same basic pattern. So these are some of the easy matches that we can do off of photographs, but here's one that I got in last month that I was just very pleased with, that I like sharing with people. If anybody gets this, you're a superstar and we want to hire you. So this is an actual case. This is all the evidence that we received. I have my thumb there for scale, and this had everything we needed. It has color, it has pattern, it had texture, which you can't get in a photograph. It was kind of loose. It reminded me of something kind of flaxen. We had a fairly good idea that the feather on the left was a contour feather, so one that came from either the bird's breast or the back. And the clincher there was the pattern, which was a black center, bordered by a stripe of bronzy brown, and bordered again by a stripe of brilliant white. Any ideas?
Bronzy brown and white.
I'll show it to you on the bird. Anybody know this bird?
Some kind of rail.
Yes, it is a rail. It is a sora.
Ah,
And this is exactly when you know that you have found your match, when you hold the feather up to the bird and you see it disappear. We get actually lots of sora in strikes, so we're very familiar with birds that you would have a very hard time seeing when you're out birding, because this dumpy, stubby winged little thing is a long distance migrant that is picking up and hauling to Central America and getting hit by planes all the time. I have never seen one when I have been birding, but I see them in bird strikes all the time. We are able to do some pretty incredible whole feather matches with the Smithsonian's collection at our fingertips. So we can really confidently identify stuff that has been chopped up in plane engines. We use anything we got, feet, beaks. Like with this black scoter, we have the adult male and we had a beak of an immature male in a bag of snarge. And we said, that looks familiar, let's take it out. Anything recognizable, we'll take it out, we'll give it a go. But one of the most important parts of being a birder, to me at least, especially when I am birding literally by parts, is knowing what I don't know, and respecting the limits of what my eyes alone can tell me. To save us when we fail, to keep me humble when the snarge is nothing more than a smear across the bow, we have Faridah and the molecular lab. And Faridah, please tell us about how you work real magic with invisible snarge. Let me know if you have trouble seeing the slides, Faridah.
Got it.
I can try and mess with things, and I will be your slide advancer.
Thank you so much. Well, welcome to the molecular identification. The folks in my lab call it the dark side of identification. I really don't know how it works either, but it just magically works. That's what it is. And next slide, Ingrid. There are 4 different methods, as Ingrid said earlier, and the DNA seem to do 50% of all the work done in the lab. That's mainly because what we get are little bits of things that are not recognizable. There is no whole feathers sometimes. That it's just, sometimes it's literally just dust that they collect, that they scrape off the plane. Sometimes it's just tissue wipes, or they scrape off the airplane. They can use anything from credit card to you name it. We get all sorts of things that are sent to DNA. And unfortunately, for some of the folks, for molecular work, it's important that you do not clean the plane with anything that has bleach, because bleach kills DNA. You can't get DNA out of it. You can't, irradiated stuff will not get DNA successfully either. But we try our very best. That's why all 4 methods are important. And the photographs also, the combined methods and morphology, they all have to work. All 4 methods have to have the same result for us to accept it, that is indeed the bird ID. And so this is next slide, please, Ingrid. Pardon me? Oh, this one just shows you a quick glance of our workload during the year. The busiest time is fall and spring, as you would imagine. That's when the birds are migrating. And it's basically the same pattern for all our clients, the FAA, the Air Force, and the Navy. And this is how we do it. It's 12. We're very busy. Like right now, we get 200 bird strikes a day. And that's a lot. And to process, it takes a little bit. Opening the mail takes a little longer, because sometimes when we get the mail, all the information is not there. So we have to do a bit of sleuthing to find out where it comes from, and all those other details that have to be connected to that bird strike. Next, please. The primary reason we can do this is because, isn't this a pretty slide of barcoding? And it's because birds and all living things have definite barcoding, which is what you see here is each genus and species have its sequence of amino acids, and each color represents 1 amino acid. And they are different, and they are distinguishable between birds, and that's what have saved our ID to do molecular work. We are not the first lab to do this. There are many labs that have done this for other animals, but we are probably the #1 lab that use it all the time. I will quickly show you how we get to this method. Next one, Lovey. This is how the original barcode looks like at the very top, the whole chromatogram. And if you stretch any one of them, it would look like the second picture. You can actually see the peaks and valleys of the actual amino acid that is being read. It can also be read as alphabet, ACTG. Those are the amino acids that we use to identify our birds. And the third way that they can do it is by color coding it. The reason to do that is it's easier to compare between birds. And I'll show you later an example of that. Next, please. Where do we get our DNA? The DNA that we work with is the mitochondrial DNA, not the nuclear DNA. And thank goodness for that, because nuclear DNA has at least 3 billion base pair. The mitochondrial DNA only have about 17,000. And we only need 647 of the 17,000 base pair to identify the bird to species. And the first top picture, the line drawing, is probably how most of us go to school and have to look at cells. Cells are now, you can even go inside the cell, you can do SEM on the cell, you can actually identify where exactly the mitochondria are. I'm born in the 50s, so I don't have those lovely photos. I grew up with a line drawing. That's what it is. And next, please. That's okay. The other half of the very cool reason we can do this is in the very, the mitochondrial DNA is presents itself circular rather than linear as in nucleosome, the nuclear DNA. And they have 37 encoding genes. And the one that we work with is this thing called CO1. We are not the first people that started this, but the early folks that tested all this, what we call primers. The CO1 is a primer, the ND2 is a primer, ND1 is a primer. The early folks that tried to do identification doing this, they tried all these primers and they found out that the CO1 is the easiest and the fastest to sequence. The protocol to make it work is relatively easy, and and you can tell quite well all the different animals apart. And also it helps that everybody in the world use the same primer, so you get the same results. So all we need again is this little bit of green is optimally, it's 647 base pair, but we can do IDs with just 500. It's such a good gene that we can get IDs with less than 647 base pair. Next one, love. So this is, so once we get the sequence, as I show you the barcode, what do we do with it? How do we get it? Because it's impossible to memorize all the barcode, even though we're pretty good at looking at the sequence and we can tell whether it's a bat or not a bat. That's because there is a series of amino acid that is in repeat that we know, that must be a bat, but we don't know what bat it is. So this is the cool thing that we have, a public and free database called the Barcode of Life that was started in 2003. And we contribute to it often. Every time we go out on a bird collection, we sequence the bird and we bank it into this database. And all data that goes into this barcode and the next one called GenBank, which is older, have to come from vouchered specimen. It can't just be any regular bird. It has to be identified correctly too. So that's what we do. A lot of people, and it's a requirement for you to write. When you write a paper, you have to bank your sequences if you are doing molecular work. Next, please. Now to our workflow. This is how we work. It's we finally this year we managed to capture in graphic how we work. It's a bit maddening, but it's okay. We open the mail and we divide it into whole feather. The word snarge, there it goes again. It's not a plural there. And then we get it, and we get photos from the mail. And if you notice, the crucial part is the middle part, that micro. Everything goes through micro for confirmation. Most of the time, we need micro for confirmation. And as the one thing that I wanted to show on this slide, more importantly, is molecular work takes a little bit of work. It will take us an additional 2 to 3 days to get an identification. And then only if it works. And if it doesn't work, we send it to micro. We have to resample again. And the ultimate goal is to be able to report to the different agencies with the correct species, genus and species. So imagine if it takes 2 or 3 days, if we have to resample to redo it again, it will take an additional 3 to 4 days. So if you have good photos and good feathers, go for it. Send us those, because we can do that in half an hour. They can do it. I don't know how to do it. Everybody else in my lab can do it. Next one, please. This is how our days start. open the mail. This is a low load mail day. we have mail buckets of mail that we get every day. We freeze them overnight just to make sure that they don't, as best as possible, we kill whatever it is in a -42 freezer. The next day we take it out. We will give it a number. Every sample that goes through our lab gets a barcode that is generated by our lab, and we scan it inside our database. After we scan each one of them, we sample it accordingly to the well number that we have, that we are using. It is a 96 well, and it's numbered 96, with the last one being negative. If the negative of that plate has a bird, it means that we have contamination somewhere, and that means we have to backtrack and try to figure out how to not get that contamination repeated. As you can see in the top photo where the gloved hand is holding the tissue, we don't need much of a tissue. We need very little, and it's in molecular, the motto is less is best. There you go. Next one, please. When we fill up the plate of 96, we happily go to the other side of the museum, which is a mighty good walk. We get our 10,000 steps a day. And with all these machines that we have an automatic DNA extraction. We have all these machines that everybody in this lab, it's a molecular lab that can have 100 scientists working at the same time. And we all work together, and which is lovely, because if we have any nagging problem, we can merely ask folks, what should we do? Or maybe they have remember those primers that I told you? Well, sometimes they have say, why don't you try a different primer or a different conditions and all this other stuff. So the first picture on your upper left is our robotic machine that extract the DNA. And one plate will take about 5 hours to extract. There is a machine out in the Museum Support Center that can extract 4 plates of 96 in 4 hours. Luckily, we have, the way we work, we don't have to do that. It's easier for us to do 1 plate at a time because there is other things to do. It's always work for Ingrid at the end of every ID because she does all the micros. And some days there's just a lot of micros because there's really nothing DNA-wise. So what we get is, after all that, we send it to that machine on the bottom right. That is the sequencer machine, and that reads all the DNA that we have submitted, and it will generate a little like a little sheet like this, purple sheet, and each one of those numbers will match that very first number that we do in the 96 plate. So we are very good at recording and tracking all our samples. And we have to do this automatically because to think about 12,000 samples can be a little bit too much. So we get 96, we run 96, we get ID for 96. And the negative, I should have shown, the negative is negative on this plate. Next slide, please. Okay, so how do you do it? Remember those, the barcode that was represented by just the ACTGs? So that's as easy as when you get that, this is a Mac thing. Oh no, PC can do it too. Yeah. You copy the sequence read, you paste it in this barcode of life, which is a free, it's a free database. And then we hit submit, and magically, go ahead, Ingrid. Yes, we got wood duck, and and it has to match. See how beautiful it is. It's 100% wood duck, and anything less than 98% we will not accept. So this is convincingly a wood duck. So this is how we get all 96 for that plate. We will get a 96 Excel sheet with the result, as we will show in the next slide. So see this slide of Excel, it has our barcode number, the real number from the example that we got, the species, genus and species, and the percentage that it presents itself when we do the sequencing, whether it do damaging or not. So the lovely thing about this is we all sit together and it's kind of like a family day. We take the case that was, after we sample, we put it back in the freezer. When we have 96 with IDs, we all sit down with pencils and we looked at whether the bird matched whether the ID matched what is in the bag. Next slide, Lavi. That's Dr. Dove. So the molecular ID in the feather lab has to be, we try to go for genus and species as best as possible. Next. It has to match 98% or better. And it has to match the location and distribution. So we have all the bird guides book out. It has to match the season and time of year, because we don't get those things immediately. Sometimes we get it a few months later. So when we get something out of place, we will usually look to see whether it is the right season. And then we visually check the remains, if there is any remains in that bag. Next, please. And if the identification are insects and other mammals besides bat, we do a microslide. It goes to Ingrid's pile. Okay, better. So this is a nice looking barcode. And then what happened is it came out Homo sapien. And the bag has feathers, so we know, and it was not a fatal case. So there must be contamination somewhere. Sometimes it's as easy as the folks just forgot that they had chicken for lunch. They didn't clean their hands, and they wipe it with the whatever they wipe it with, and put it in the bag. And our ID is only good for what little tissue that we sample. So there could be a bird in there, but what we sample look logical to us. That is the piece that we sample. It was Homo sapien. Sometimes we have to take the tissue because there is no tissue that's obvious. So when we take the tissue, it could jolly well be from Homo sapien. So when we do sit down, we say, no, that is certainly not correct. So what do we do with a contamination? We look at the evidence to see if there's a good reason why it is not what we think it is. And then we send it to Ingrid for micro identification. And if it's not possible and we need the answer to be to genus and species, we will try again. We will resample and re-extract. We will try a different set of primer, and we will use a different dilution for PCR. PCR in the lab is the beginning part of what you do molecular work. What it does is it makes millions and millions of copy of this region that we want, the CO1 region, the 647 base pair. If you already stoke it with so much DNA, it overwhelms the reaction and just say, I can't do it. I can't do it. I'm not going to do it. I'm not going to give you the sequence that you want. So sometimes we have to even dilute the DNA because the machine is rather efficient. We have to even sometimes dilute the DNA that we extracted to 1 in 100. That's how little it can be, because we're making millions of copy from that one primer. Next, please. This is our sad story. Hit it, Ingrid. We cannot tell these guys apart. The white-headed gulls. We don't know what to do with the white-headed gull. We get Larus, we get the species, but all of them are all the top hits, top 100 hits. Even if you manage to look at the top 500 hits, there are all different kinds of white-headed gull. It is as confusing as this photo is. But luckily, the people in my lab know how to look at feathers. So they can sometimes get it to species by looking at the feathers, taking it out to the collection, looking at where the location of the strike is, and try to match it as best as possible. Next, please. We have attempted to try and look for the species for a white-headed gull. This is the problem. It's not really a problem. Right now, we don't have enough personnel to do it. We have done in the past, and it was a very, very exciting work done by a postdoc with Carla. And we have to use 6 different sets of primers, 6 different protocol in the lab. And the best you can get is the highest probability of a species, 60% best. So we're still not there when it comes to the gull. Black-headed gulls not a problem, but white-headed gulls is still a problem for us. Next, please. Okay, let's talk about the positive things, what we can do with molecular protocol. We can tell apart using a different primer. Remember the CO1 I mentioned earlier? When we do a CO1, we get horn lock, but we don't know whether it's a streak horn lock or a regular horn lock. And in the Northwest, they have designated the streak horn lock to be on the Endangered Species Act. So when we do get a horn lock from a certain area in the Northwest, we do it again. We already have the DNA. We don't have to re-extract. We have the DNA. We run it with a different set of primer, a much longer primer. It is about 1200 base pair rather than just 647. And you can tell apart the streak horn lock from the horn lock. So this has saved us quite a bit. And we have managed to do, to be able to call out the streak horn lock. And it's Aromophila alpestris trigata. They just added another word in the genus to make sure that it is to designate it as a streak horn lock. We can also tell the diving ducks apart because when we get it with CO1, it is all the ITHEA, all the overlapping barcodes of diving duck. But with the ND2, you can tell them apart very nicely. Okay. And as for number C, using the CO1 and a phylogenetic tree, that's what molecular people love to build trees, phylogenetic tree, to see who's related to who by what percentage. We can tell certain species of warblers, thrushes, buzzards, nightjars, and many more. We are always, this list under C, we are constantly working on it to see whether we can tell birds apart, much better down to species, especially when we get birds from overseas. We get strikes quite frequently from overseas, not only from the American bases, but from commercial airlines, Singapore Airlines, and all those other folks will send us, and also engine manufacturers. They will send us when they pull out the engine, they will also send us the snarge. And the other cool thing, the simplest of all molecular work that you can do is to tell them whether they are male or female. It's as easy as running another set of PCR, run it on a gel, 1 line male, 2 lines female. And this is especially important for damaging cases because they would like to know whether how heavy the bird is. And there is a big distinction in the weight of the male and female. And it's always welcome if it's a female. If it's a male, they always say, oh, okay, I wish it was a female, but it wasn't. And we have this, yeah, we can tell genetic, the genetic sexing is very, very easy and cool. Next one, please. Everybody know 1549 US Airways. We've been doing this forever, but nobody know that we do this with a full load, with a full staff until this happened. When this happened, I happened to be working there already. The phones were ringing and we were told by the director's office not to answer any calls because they knew he mentioned that they hit geese. And so they knew that the only place that might be sent, because it was the FAA was taking over the case, that was going to be sent to us. So reporters start calling. So we couldn't answer the phone. The next few slides are the pictures that we got from 1549. And we all remember this photo. It's being dredged up from the icy water. By the way, we got all sorts of stuff. They sent us 70 samples and none of them were birds. Some of them were some weird bacteria, that parasites living in the water of the Hudson. It was, we even got medical waste and it was really, it's scary to think out of the 70 samples, I think only about 50 that we can use. The other 20 were weird things that I don't even know what they are. I have to look up what they were. And next slide, please. And when they took it out, and that's our guy, lower right, His name is Mike Beezer. He's our connection to all he goes to and investigate it and pull out the best looking sample for us because he knows it's kind of critical to know. If he can get feathers, he will certainly collect feathers. And the next slide, I'm just going to show you a little bit of a test case. Remember, I mentioned that some of the chromatograms can be presented as color-coded. So we were told, this is from 1549, we were told it was a goose. Sully said it was a goose. And what we got was also goose. From the initial molecular work, we also got goose. But we don't know, in that area in New York City, there's two kinds of goose, a Canada goose and a cackling goose. So when we did all that, the slide before, Ingrid, yeah, and on this portion of 647 base pair, if you notice the color shift, there were 13 different color shifts when we compared the results. And then we knew that it is certainly not cackling goose because when we blast it in and make a phylogenetic tree, as Ingrid will show next, we see our unknown over here is definitely a Branta canadensis and not a cackling goose. So that's how some of the ways that we use DNA molecular work. It's always fun. It's never boring. And we're only talking about ACTG. That's it, you know. Well, thank you so much for listening to the dark side of identification. That's it. Back to Ingrid for microstructure, her specialty. She's incredibly good at this.
Thank you, Faridah. I'm going to wrap up by talking about the method we wrap up with. This is the cleanup method. Our microscope works when all other methods fail. Microscopic identification of feather fragments is an especially important tool when we work catastrophic cases like the miracle on the Hudson, when the aircraft is at the bottom of the river, when the aircraft is burned up and there's no DNA left, or when it has crashed in a hard-to-reach area and the snarge has remained inaccessible for so long that there's feather dust. Microscopic ID is our lab's specialty, and Dr. Carla Dove, who's our lab's leader, is the world's foremost expert in this. So we can do this because the body feathers of birds have a secret hidden signature built into their structure. When we look at the body feather of a bird, it's divided into two parts. There's the pennaceous part, which is the part that we're using in our whole feather ID. It has color, it has pattern, it forms a water-resistant protective covering over the bird's body. And then there is the plumulaceous part. That's the soft, fluffy down that keeps a bird warm. The microstructure of the pennaceous part of the feather is the same between all birds because it has to zip up like Velcro to form a feather vein. So you can see on the top, the little hooklets. That picture in red and blue is actually a very zoomed in microscope image of Velcro. It looks almost exactly like Velcro. But the downy portion of the feather, it just has to be fluffy. It is free to vary, and it looks different between all the major groups of birds. All we need are a few fragments of that downy feather, and we have got ourselves as good as an autograph from this bird.
That's right.
So we can identify to order or family, and with additional evidence, maybe from the molecular lab, thank you, Faridah, we can get to the species level. But microstructure can get us a very solid identification at a higher taxonomic level. These are some examples of what different downy microstructures look like. So the feather think of a feather like a Christmas tree. It has a central shaft, which is the trunk. It has branches that come off, and then there are leaves that come off the branches. And each one of those leaves is a string of individual cells. Those individual cells can be triangular, they can be star-like, they can look like beautiful little black teardrops, they can be rings, they can be spines. They might even have strange little fringes called villi sticking off them. And here is what that down actually looks like under the microscope. This is the down of a bird from the order Accipitriformes. So this is a bird of prey, and our hawks and our eagles have downy feathers that are very simple, almost like bamboo. They have sort of a stippled brown pigment throughout them and tiny little spines at each of the little, you can see these little swellings where the cells join to each other, and little tiny spines sticking off them. But a bird from a different group is going to have down that looks very different. Things start to get weirder when you look at our ducks and geese, our Anseriformes. Their down is very simple at the base, but out at the ends of the leaves, they have triangular shaped swellings that are very distinctive under the microscope. And we have found fragments of these little triangular down pieces in Viking pillows that are hundreds and thousands of years old. They preserve very well. The down of a perching bird looks different still, with these, I call it the black pearls on a string. As you can see, the upper image here on the right is through a transmitted light microscope. That's what I'm using, what you might have used in a lab class in college. You put it on a microslide and the light shines through it. The feather itself is transparent, except for the little drops of black pigment trapped within the feather. And at the bottom, you can see what that looks like under a scanning electron microscope, which is another tool we have at our disposal. We can coat our sample in gold. Don't ask me how to do that. We have a guy who does that for me. And then you can image it with a beam of electrons. And it is easier to see here the weird little fringes that come off the base of each one of these sort of leaves on our tree, like little insect antennae with knobs at the end. And whenever I see that, I know I've got a perching bird. And then even wilder things, I just think that hummingbirds have the most beautiful down of any bird. These beautiful dark stars against a light sky. And they even have asymmetry in the feathers. So all of the stars line up on one side, and the other is just sort of very simple and bamboo-like again. And if you ask me why it grows like that, I have no idea, because there's still a lot to learn about feather microstructure. There are even more unique downy feather structures I could show you. But I couldn't forget to give my friends, the bats, a shout out. We get a lot of bat strikes as well as bird strikes, especially around this time of year, as bats are also migrating. And I actually started my career as a mammologist, so I have a soft spot for bats. The individual hairs of bats also differ in structure according to the different families and genera of bats. On the left, you can see the free-tailed bats. We call this the punk rock look. They're very spiky and I don't know, fierce looking. And on the right, we have hair of a Vesper bat, something like a little brown bat or a big brown bat. And they kind of have leaf-like scaled structures, keratin, just like my hair is made out of. Often when a sample comes back from the DNA lab with an identification of insect, I say, oh no, I want to double check that, because when I look at it under the microscope, I might find a bat hair, surprise, mixed into what the DNA identified as a moth. But I could talk about microstructure all night. I don't want to keep you too long. As Faridah explained, we use all the methods of identification at our disposal together in combination with each other. So each one of our methods supports and holds up our identification. We take into account the facts of each case, and we even use eBird and our field guide library. We like to use eBird to do a sanity check sometimes, just to make sure that, has somebody recently seen that bird around the Dallas-Fort Worth airport? That's a strange identification. Do you think it hit actually on takeoff somewhere else? And we discuss these things. We take into account our diverse expertise among the members of our team to arrive at each one of our identifications. But to what end?
Over here.
Prevent things like this from happening. And even very small birds can produce a surprising amount of damage. We had a damaging hummingbird strike last week because little birds just seem to have a magnetic draw towards the most expensive parts of the plane. We cannot stop bird strikes if we do not know what birds are being hit. All of our identifications are entered into databases that are maintained by the Federal Aviation Administration and the US military. And the reported number of strikes to civil aircraft have been consistently increasing over time. If you ignore that little dip in the pandemic when nobody was doing anything, it's looking for the past 2 years, like the numbers are heading right back up to our 2019 levels, which was the highest number of bird strikes we'd ever had. And that makes sense because more people are flying commercially today than at any other point in history. But we also know that the proportion of damaging strikes that put people at risk is slowly decreasing due to the hard work of people analyzing and using this data to keep you safe. Most major airports at the United States employ airfield biologists who work directly with our data to make their airfields safer. The vast majority of strikes happen at takeoff and landing, and airport biologists use the species identification to modify airfield habitats to make them less appealing to the kinds of birds that are being hit. For example, if an airport is suddenly seeing large flocks of mourning doves being hit, they're going to search their airfield for what is attracting those doves. They're going to remove food plants so that birds no longer have a reason to feed in a dangerous place. If gulls keep hitting planes and there's an open landfill at the end of the runway, which is something that happens a lot, airport managers have the data that they need to justify getting that landfill covered and hopefully encouraging those gulls to move somewhere else. And they also figure out what birds aren't a problem. Maybe there's going to be a big push to remove crows, but it turns out crows almost never get hit by planes. They're just far too smart, and we get very few crow strikes each year. They can direct their energy to where it's going to be more useful. Airfield biologists also employ hazing to scare birds away with loud noises. Dogs, even other birds, employing a falconer to scare away prey species. And different species are going to react differently to those different deterrents. So we need to know who we're targeting to make our interventions effective. And this is just something I'm fascinated with. All of these new technologies that are available to airport biologists that in combination with bird strike data are in the process of transforming our understanding of airports as bird habitat. Bird radar that can detect individual birds is a thing that exists. This is an image of bird radar in action. These little yellow dots with the red tracks flowing behind them are flying over JFK Airport, and they are a flock of brant that are being picked up on the airport's bird radar. When airports know what their problem species are and can track their habits, their movements, intuit what is attracting them at the level of microhabitats on the airfield, bird deterrence strategies become even more effective. And we can also learn to live with birds and practice avoidance by altering our behavior in turn. New tools that are available to you as birders, like Birdcast, are being investigated to potentially alter flight schedules on nights when we're sharing airspace with thousands of migrants. There's so much new research that is happening about how to keep people safe, and it all begins with knowing who's out there interacting with us in the wild and tracking strikes that we wouldn't know about if we didn't get a little envelope of snarge in the mail. We like to say that we don't want our children to be playing on airport tarmac, and neither do we want birds to be playing there, because we love birds. We are fascinated by them. We are birders ourselves, and every day we are working to keep both birds and people safe. We wanted to thank you for the opportunity for having us give this talk. I don't know how much time we have left. I wanted to leave time for questions. I have more random facts and some interesting cases appended to the end if people have an appetite for more, but I don't want to keep you from your meetings. I'm also happy to open it up to questions. Thank you so much for having us.